interphase, there's 3 stages. The first one is G1, so that's when the cell begins to grow and it expands, and the centrosomes also begin to replicate, and it's getting ready for the DNA to replicate too. So it could go on for a few days or weeks or years, and once it's reached a size that it has to be, it'll go on to the S phase. Next is the S phase, which is synthesis, and this is when all of the chromosomes will replicate, so there'll be 2 times the amount of chromosomes that there was before. And the cell will continue growing in its size, and so will all of the number of proteins and enzymes will replicate too. And then after this, it goes to G2. G2 is the intermediate phase to make sure that the cell is ready to go into the next part of the cycle and to split into 2 cells during mitosis. So it just checks to make sure that all of the cellular components have been properly duplicated. The next part of cell division is mitosis, which has prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis. So we're looking at prophase right now, which has the nuclear envelope still attached with our chromosomes in it, and the centromeres have bipolar spindles that are beginning to develop on the outside of the cell. Now we're on to prometaphase. So the nuclear envelope has dispersed all around, and now we have the kinetochore microtubules forming with our, and as they align, it's going to force these so that they'll move. So next is metaphase, and the centrosomes have aligned at opposite ends of the cells, or of the cell, and the chromosomes are moved to the center of it, and they're aligned at the center. So between each mitotic spindle is where you'll find the centrosomes. The next phase is anaphase, and that is, you can tell that it's anaphase by the separation of the sister chromatins, and the centromeres will begin to move apart by the increase of the microtubules, and this only lasts for a few minutes before it moves on to the next phase. Because we have an animal cell, cytokinesis, which is this step, goes right after anaphase, and then it'll be followed by telophase. So this is our cleavage furrow, which is the crack in between the 2 cells that are formed, and eventually it's going to separate completely in 2. And we have, oh no, where'd it go? And we have our nuclear envelope with our chromosomes inside and our centrosome pairs on the outside. The next phase is telophase, and we have the nuclear envelopes beginning to reform around the chromosomes, and our cell has elongated, and then eventually the spindles are going to start disappearing. So now we're at the G0 phase, which is the resting phase, where the cell is not dividing and it's not preparing to divide. It's seen as an extended G1 phase. So our cells have been completely separated.